Hey everyone, have you ever wondered that how cells divide? So today we are going to talk about that. So let's get started. So let's talk about cell division first. So cell division is a process by which cell splits into two new cells, and it is of two types, that is meiosis and mitosis. In meiosis, a diploid cell is converted into four haploid cells, and in mitosis it is converted into two diploid cells. So let's talk about mitosis in detail. Well, mitosis, as I already told you, that it produces two identical daughter cells. It mainly occurs in somatic cells. It maintains the chromosome number, and there is of course no genetic variation in mitosis. Now the stages of mitosis. Well, after interphase, there were four stages of mitosis, that is prophase, metaphase, anaphase, and telophase. So let's talk about prophase in detail. The beginning of prophase is marked by appearance of condensed chromosome, each of which consists of two sister chromatids. And you can see that the condensed sister chromatids are then held together by a point that is known as centromere, which separates and move to opposite side of nucleus. There, they serve as two poles of mitotic spindle, which begins to form during late prophase. Now in metaphase, the mitotic spindle fully developed, and you see that the centrosomes are at opposite pole and the chromosome lined up at metaphase plate, and each sister chromatid is attached to spindle fiber originated from opposite poles. Now after metaphase, there is anaphase, in which you can see that the sister chromatids pull towards the opposite pole. And lastly, there is telophase, where the chromosome arrives at opposite pole and begin to decondense. You can clearly see from this diagram also after interphase the four stages and how these four stages were leading to the formation of two diploid identical daughter cells. Now let's talk about the second type of cell division, which is meiosis. Well, in meiosis it mainly occurs in germ cells. It produces four haploid cells, and yes, there is genetic diversity. Now, the meiosis has two sequential rounds of cell division, that is meiosis 1 and meiosis 2. So you can clearly see from this diagram that there were four stages, that is prophase, metaphase, anaphase and telophase, both in meiosis 1 and meiosis 2. But in meiosis 1, the prophase 1 is further divided into five steps, that is leptotene, zygotene, pachytene, diplotene and dikinesis. This mainly is related to the crossing over between the chromosome. So in the meiosis 1, there is mainly reductional division, whereas in meiosis 2, there is mainly equatorial division. Now let's do a quick comparison between meiosis and mitosis. Well, the difference is mostly on the basis of the four stages in both mitosis and meiosis. So you can see clearly how in mitosis there is formation of two diploid cell and meiosis there is formation of four haploid cells. So this is all regarding mitosis and meiosis. So if you found this video helpful, do like and share, and don't forget to subscribe to my channel.